Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing National Entrance Screening Test NEST 2022 Session 2 question paper. I will be discussing only the biology section. So coming to the first question. The path of transmission of electric impulse in a nerve cell is. So it's a direct question. It is from dendrite, then cell body, axon and then nerve ending. Then consider two spherical cells P and Q with radii 1 micrometer and 0.01 millimeter. Choose the statement that is correct regarding these cells. Now I have done the calculation in the next slide. You can go through that. So 1 micrometer, we are converting it into millimeter, that is 0.001 millimeter. Now we know that it is a sphere. So the surface area of sphere is 4 pi r square and volume of the sphere is 4 by 3 pi r cube. I have done the calculation, that is for P I have found out the area and the volume. For Q also I have found area and volume. I have also found surface area by volume, that is 3000, and here for Q also area by volume I have found, that is 300. Now which is having as per the options. Now as per the option 2 is correct, because P by Q is 3000 divided by 300, that is 10 times. So P is 10 times more, having more surface area volume ratio than Q as per option 2. So the answer here is option 2. Why option 1 and option 3 are wrong? I have done the calculation here. If you see option 3, they will be telling that area will be and volume will be doubled. But volume will not be doubled because volume is directly proportional to r cube. So it is not doubling in case of volume. So option 4 is wrong. Why option 1 and 3 are wrong? I have given it here. Option 2 is the right answer. You can go through the calculations. Then next one. Deoxyribonucleic acid is a polymer composed of the nucleotides deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxythymidine triphosphate and deoxycytidine triphosphate. Two such polymer strands are held together in a DNA duplex. From the following options, the correct representation of the typical Watson-Crick model of the DNA duplex structure is. So as per the Watson-Crick model, the nucleotides of the DNA are joined in a polynucleotide strand by phosphodiester bond that connect the 3' carbon atom of one nucleotide to the 5' phosphate group of the next. So this is the 5' phosphate. It is connected to the 3' carbon atom of the other nucleotide. So here also the phosphate, that is 5' phosphate, is connected to the 3' carbon atom of the next nucleotide. Then you have got a this is I think adenosine, adenine. This is adenine, that is base pair to thymine in the second strand, and here guanine is paired with cytosine in the third, in the next strand. So ATGC pairing is clearly seen. Then the nucleotides are attached, the bases are attached to the C1 of the sugar. So that is also correct. In here also they are attached to C1 of the sugar. So that also correct. So this figure is more accurate. Whereas if you see this figure, it is not to the this is not the 5' phosphate. This is C1 of the sugar, not the 5'. So this is not correct. If you see here, this guanine is paired with another guanine in the second strand. So that is also not correct. And here if you see, the bases are attached to the C1 and it is on the other, the orientation is not correct. Here also, here it is correct, but here the orientation is not correct. Sugar bases should be on the sugar phosphate backbone is for structural representation of the DNA, so it should be on the outside, and these bases, they should be on the inside of the helix axis. So that is not correct. Now next one. In a family, the third child showed Down syndrome, but neither of the parents nor siblings of the affected child manifested symptoms of the syndrome. The possible reason for Down syndrome only in affected child is, so it is due to chromosomal segregation defect during ovum development in the mother. So what is this chromosomal segregation defect? So in normal meiosis, you will see that first the chromosome separate, then the chromatid separate, and upon fusion with the sperm cells, you will get a zygote having 46 chromosomes. But in the case of Down syndrome, it is due to trisomy of the 21st chromosome. Here, during meiosis 1, the chromosomes do not separate. They are as such taken to the only one cell and not being distributed to two cells. So one cell will be having this particular chromosome and the other cell will not be having that chromosome. So once upon then meiosis 2, the chromatid separate, but this cell will be the one which is defective cell, that will be having two chromatids. So upon fusion with sperm cell having one chromatid, you will get three chromatids in the zygote, and this is trisomy. So trisomy of the 21st chromosome results in Down's syndrome. So it is happening because of segregation defect in meiosis 1 of the mother. The next one, primers P, Q, R and S have binding sites on the template DNA as depicted in the figure below. In a polymerase chain reaction using all four primers together, the possible number of amplified products of different sizes is. So here P and S will give you one size amplified product. Then P can be the forward primer and R can be used as the reverse primer, and you will get another set of amplified products of different size. Similarly Q and S as well as Q and R. So totally how many? 4. You will get 4 different amplified products of 4 different sizes. Then the pedigree shown below represent the inheritance pattern of X-linked recessive color blindness trait. In the pedigree, circles represent females and the squares represent males. Filled spaces indicate affected individuals while unfilled space indicate unaffected individual. Individual 3-1 exhibit Turner's and also manifest color blindness. Genotypes of individual 2 and 3-5 respectively would be 2-3, that is this individual and this individual. What would be the genotype? So this is the pedigree and they have asked for 2-3 and 3-5. Now this individual is, that is the father, is having color blindness, mother is not having the trait. So this is the case with father, this is the mother, and upon crossing you will get XCY, where XC is color blindness and Y chromosome, and X is normal chromosome without color blindness. So you will get XC, XY. So 3 is not having the defect, so that means she is a carrier of the disease, not having the disease, but carrying, not having the disease but carrying the diseased gene. So 2-3 will be XC. Now coming to 3-5, that is from here. So this female child could be XC and it's crossed with a male which is normal. So XY. So you will get offsprings that will be XX females who are normal, that is here Y, or they can be XCX also, that is because they may be carriers also, because the mother is having a heterozygous genotype. So the this 3, 5 could be either XX or XCX. So the answer here is option 1. Then following are statements about different stages involved in plant development. Conversion of root apical meristem to root cap cells. Conversion of mesophyll tissue to callus tissue. Formation of xylem from vascular cambium. Now before coming to the option, let me tell you what is differentiation. That is a process through which meristematic tissue undergoes permanent change to form specialized cells in the plant body. That is called as differentiation. And what is de-differentiation? The phenomenon of the reversion of mature cell to the meristematic state leading to the formation of callus is called de-differentiation. So first one is the first differentiation, that is conversion of root apical meristem to root cap cell. So the meristematic tissue here undergoes permanent change to form specialized cells of the plant body. So it is differentiation. Then conversion of leaf mesophyll tissue to callus tissue. Here the reversion of mature cells, that is leaf mesophyll cell, to meristematic state, that is leading to the formation of callus, it is called as de-differentiation. So 2 is de-differentiation. Formation of xylem from vascular cambium, that is differentiation. That is the cambium is undergoing change to form xylem. So the answer here is differentiation, de-differentiation and differentiation. Then next one. Choose the correct set of molecules from the options below which are required along with water and carbon dioxide to drive photosynthesis in dark condition in green plants to produce sugar. So you need ATP and NADPH. Answer is option 1. It's a direct question. Then, in a population undergoing logistic growth where finite resources determine population growth, the growth is highest when the population size is half the carrying capacity. Answer is option 2. Then next one. In order to study the effect of external cues on circadian rhythm, the spontaneous wheel running activity of flying squirrel in total darkness was recorded over 24 hours for several days. The results are shown below. Active period appear as dark broad lines. That is, these lines are active periods. Before these records were taken, the squirrels had been kept on a regular cycle of 12 hours light and 12 hours dark as indicated at the top of the figure. So this is the activity pattern when kept in normal 12-hour light and 12-hour dark cycle. And this is because for activity record below are from continuous darkness. That is this. Based on the result, the correct statement is: there is a slight delay in the period of activity each night as the experiment proceeds. That is not correct. The pattern and periodicity of the circadian rhythm in the squirrel is maintained throughout the experiment. It is not so. So this second option is also not correct. The natural circadian rhythm of the squirrel is approximately 12 hours. It is not 12 hours, it is 24 hours. So that is also not correct. On removal of external cues, the natural circadian rhythm is retained for the initial 24 hours. That is correct. So the answer is option 4. Then next one. On an island, black-bellied seed cracker birds feed primarily on seeds of 2 species of sedge plant. These seeds are similar in size but differ dramatically in their hardness. The distribution of bill size as measured by the width of the lower section of the bill, that is mandible, for an adult population of these birds is represented below. So what type of selection is this? That is the question. So you know that it is a disruptive selection. I have already explained disruptive selection in the previous question paper. So disruptive selection is it produces population that has 2 extreme versions of the trait. One is the dominant phenotype and the other will be the recessive or the phenotype. That is, it may be either short or it may be tall. The intermediate height will not be observed or will be very, very rare in the case of disruptive selection. That is, extreme phenotypes are selected and intermediate forms are disseminated. So the answer here is disruptive selection. These are the extreme forms. This is one extreme and this is the other extreme, whereas intermediate forms are not there. Then next one. In a nutrient poor habitat under study, it was observed that pine trees depend on mycorrhizal fungi associated with the root system for nutrient acquisition from the soil. The mycorrhizae in turn depend on the pine tree for energy in the form of carbon. Voles, a kind of rodent, were found to be present in the habitat, and their association with the pine tree and mycorrhizae is depicted in the schematic below. Based on the given information, choose the correct statement. So, tree with rootlets having mycorrhizae, the fruiting body, that is ectomycorrhizae, which is called the truffle, is consumed by the vole, and the spores, fecal pellets with spores, are disseminated, which again infects the root of the pine trees. So, based on this given information, choose the correct statements. The three species involved are all in direct, obligate, mutualistic relationship with each other. Mycorrhizae show mutualistic association. Okay, so first option is not correct. If the young pine tree invading the field failed to acquire mycorrhizal symbionts, the pine tree population could decrease over time. That is correct. If they don't form symbiont, then they will be starved of nutrients. So second option is correct. A marked decrease in the vole population would have a negative effect on the growth of new pine trees throughout the habitat. Because vole consumes the truffle, that is fruiting body of ectomycorrhiza, and the spores are released along with the species. And the spores infect the rootlets of the pine tree. So when the vole population decreases, the amount of spore released will be decreased. And this results in decrease in the amount of mycorrhizae attacking the new tree. And the trees will not be able to absorb nutrients from the soil, and their population will eventually decrease. So it is having a negative effect on the growth of new pine trees throughout the habitat. So option 3 is also correct. The mycorrhizae share a parasitic relationship with the tree. It is not parasitic, it is mutualistic or symbiotic relationship. The next one. A mutation in an autosomal gene, TUM, causes benign tumor in a dominant manner. T and small t are the two alleles of the gene. A mutation in another autosomal gene, MAT, that is alleles capital M and small m, determine conversion of the benign tumors caused by the gene TUM to a metastatic one in a recessive manner. A couple, Mr. Y and Mrs. X, both heterozygous for the genes TUM and MAT, have a child together. Based on this information, choose the correct statement. It's a very simple Mendelian genetic problem. So here there is a gene, autosomal gene called the TUM, that causes benign tumor when it is found in the dominant manner. That is capital T. If it is there, then it is due to the gene for benign tumor, that is TUM. And there is another gene that is called as the MAT, and it determines the conversion of benign tumors to metastatic one. That is, metastatic means cancer causing. So these mutation in the another autosomal gene, that is MAT, in a recessive manner. So if MAT is present as small m, that is the recessive manner, then there is conversion of the benign tumor to a malignant one or metastatic one. So these are the phenotypes of the parents X and Y. They are heterozygous, they have told. So these are the phenotype, these are the gametes produced, and this is your Punnett square. Now after the Punnett square, if dominant T is present, then benign tumor, and recessive M, that is small m, then it results in metastatic condition. So here they have asked how many will be having no tumor. So small t, no tumor. Capital M, it will not be metastatic. So it will not have tumor. So 1, 2, 3 and 4 will be non tumor. So 1/4 of the child will be having no tumor. Then 9/16 of the children, they will develop benign tumor. That is this condition, capital T and capital M, not small m. Only if small m comes, then it will be metastatic. That is over here. If you see this condition, it is metastatic. Here also the tumor develops and it becomes metastatic. That is malignant tumor, not benign tumor. Now how many will develop benign tumor? So it will be 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9 out of 16 they will develop benign tumor. Then next one, 4 ecological pyramids 1 to 4 are shown below. The correct statements about these pyramids. 2 can be energy pyramid for temperate forest if secondary carnivores would have greater value than primary carnivores. That is not. Then third represents biomass pyramid while 1 represents an energy pyramid for an aquatic ecosystem. That is correct. If 4 represent a biomass pyramid for a terrestrial ecosystem and if the producer has a total of 75,000 kilocalories of energy, then approximately 750 kilocalories of energy would reach the secondary consumer. That is correct because that is based on the 10% law. When energy is passed on from one trophic level to another, only 10% of the energy is passed on to the next trophic level. So from 75,000 kilocalories, that is producers, 10% of it will be passed to the herbivores. That is 75,000 into 10 by 100, you will get around 7,500. Now, so out of 7,500, the amount of energy that reaches the secondary consumers will be 7,500 into 10 by 100, that will be 750. That is for secondary consumers. So that is correct. First and second could represent number of pyramids for a grassland ecosystem, a tree ecosystem. That is also correct. Then next one. Schematic P shows restriction enzyme site mapping on a DNA fragment containing gene small w that codes for protein capital W. Schematic Q shows a plasmid-based vector map and restriction enzyme recognition site sequences in the multiple cloning site. The oblique line in the sequence next to each restriction enzyme name show the cleavage site of the respective restriction enzyme. If the goal of an experimenter is to clone the gene small w into the vector to express the protein capital W, the correct restriction enzyme pairs from the options below that can be used to digest the DNA fragment as well as the vector is or are. Now this is your gene of interest, this is the vector, this is the multiple cloning site of the vector. Now it has got EcoR5, HIN3, BAMH1, APA1. And your gene of interest, that is gene W, is also having EcoR5, HIN3, BAMH1, all these restriction sites. So you cut the gene with the particular restriction site, with a restriction enzyme, and with the same restriction enzyme combination, you have to cut your vector also. Only then you will be able to ligate this particular gene into the multiple cloning site of the vector. So that is what they have asked. Now we will see the options. So coming to the first option, that is HIN3 and APA1. HIN3 is over here, APA1 is on the other end. Similarly, in the case of the vector also, HIN3 is on the left end and APA1 is on the right end. So naturally it can be ligated to the vector. Now HIN3 and EcoR5. HIN3 and EcoR5 occur on the same left hand side on the vector. But in the case of the gene of interest, you can find HIN3 site here, an EcoR1 site, EcoR5 site here, as well as an EcoR5 site here. So it cannot be used for ligating. Then echo R5 and APA 1. Echo R5 is at this end, APA 1 is on the right end. Similarly, echo R5 is on the left end and APA 1 is on the right end. So naturally it can be used to ligate. Now BAM H1 and echo R5. BAM H1, in case of the gene of interest, it is on the right side as well as on the left side. But in the case of vector, BAM H1 is on the right side and echo R5 is on the left side. So it cannot be used for ligating. The next one. A tree depicting the evolutionary relationship between animals A to H is shown below. With respect to the evolutionary tree, the correct statement is or are. If E is lungfish, G and H could be birds and mammals respectively. If E is lungfish, G and H could be birds. Yes, amniotic eggs and mammary glands. Naturally, it is birds and mammals. So, one is correct. C and D could be flounder, fish and salamander respectively. They are spiny rayed fins. So, salamander doesn't have spiny rayed fins. So, option 2 is not correct. Then A and B could be prawn and crab. Exoskeleton and molting. So, A and B could be prawn and crab. That is also correct. F could be ants. It cannot be ants because ants don't have amniotic eggs. So, F is not correct. So that's it. Thank you for watching my channel. Please do like, share and subscribe.